So it is my pleasure today to introduce Dr. Olga Botvinnik. And Olga is a bioinformatics data scientist at the Cz Biohub in San Francisco, California since 2017. And she's doing currently research at the interface between single-cell transcriptomics and machine learning. Before that, Olga did her PhD at the University of California in San Diego. And currently, she's also involved in quite a few volunteering projects, fun ones like cupcakes and coding we've heard at the Cz Biohub. And also she's currently involved in the NF Core project for reproducible bioinformatics pipelines, in which we had the chance to meet. And yeah, I'd like to thank Olga for joining us today. And we are really looking forward to her talk on single-cell transcriptomics for 99% of animal species without reference genomes.
Thank you so much for that kind introduction, Gisela. Looking forward to sharing the science I've been working on with you. So I figured I'd start with what is Cz Biohub, because it can be a bit enigmatic for people. So first I want to underscore the difference between Cz Initiative, which is a sister organization to Cz Biohub. You may have heard of CZI for funding for scientific projects. Science is one of the things they do. They also have efforts in healthcare, in education, and in criminal justice and social justice reform within the United States, but also fund many grants internationally for scientific projects such as open source software. NF Core and Nextflow have received one of those. I was not involved in that, and other imaging projects. Cz Biohub is a nonprofit biomedical research organization based in San Francisco. It was founded in 2016 with a very generous gift from Priscilla Chan and Mark Zuckerberg. It's based in San Francisco next to the University of California, San Francisco, UCSF campus, and has satellite facilities at Stanford and Berkeley. This is the first time that UCSF, Stanford, and Berkeley have actually collaborated together in an official capacity to accelerate science. And the main goal of Cz Biohub is to promote scientific research to accelerate that can cure, prevent, manage, or treat all disease by 2100. Ambitious goal, but if you think about 100 years ago, vaccines were not commonly used or even invented yet. And those provided a huge advantage to public health. And just to briefly describe some of the efforts at Cz Biohub, first is the investigator program in the first third, which is about 100 investigators at Stanford, UC Berkeley, and UCSF who collaborate together through Cz Biohub. The other third is technology and platform development. That's where I am. I'm on the data sciences platform. There's also engineering, genomics, and imaging. And finally, there is the intramural research program. So I collaborate with people in the infectious disease initiative and in the Cell Atlas initiatives. So that's enough about Cz Biohub. A bit about me. So I'm Olga. I build stuff. I build individual tools, which then I combine into pipelines, which enables me and others to see the data in a new way. And the goal is to crack open new fields of biology with these new tools. So I want to tell you about a few tools I've been working on, and I will first speak in an analogy. So let's say we have two books in different languages. Are they the same book? How could you tell if they are the same? Well, for each language, you would need at a minimum a dictionary, the index of all possible words, and a definition of what all the words mean, and a per word translation from a word in one language to the word in the other language. So a mapping of the words from one to the next. What I'm asking, and what I will argue, is that you don't need a dictionary. You don't need a definition. What if you didn't have a dictionary? You didn't have a definition list. Could you tell if the books were the same, had the same content, if you just had the per word translations? That's all you had. So let's continue this analogy. So the book is like a transcriptome. So if we think in a very generic sense, a book is a subset of all possible words in the dictionary. Maybe the book invents some new words, but it's largely a subset. And that's like the RNA. It's a subset of the all possible words that could be on, all possible genes that could be on. So for a dictionary is kind of like a reference genome. We can analogize it as a list and simplify it as a list of all possible genes. The word in its definition could be like the gene and its annotation. What does that gene do? Now the per word translation I described before of mapping of one word to the next, this is like mapping equivalent, also termed orthologous genes from one species to the next. And in this talk, I will greatly simplify the problem to only focus on a one-to-one mapping of genes across species. And there's much more to it, but we'll just focus on the one-to-one mapping. Now, what's the issues with this is that this first dictionary step is very challenging to obtain. It's now much simpler. It's very accessible to obtain a transcriptome, this first step, the books of different species, but creating a high quality dictionary, a high quality reference genome is much more challenging. And many of the existing reference genomes are more like a stack of pages than a full dictionary. So let's dive into this a little bit more. So on Earth, there's about 10 million animal species predicted to exist on Earth, and a few thousand of them have genomes that are submitted to the American National Center for Biotechnology Information, NCBI. And this has that has a submitted genome. Now, if we want to look at high quality genomes, we can turn to the European Ensemble database, looking at genomes that have not only their initial sequences, but also gene annotations with the definition. So definitions of the words is in the analogy. Then we go down in order of magnitude. About half of those with definitions have some translations to, we'll call it English. So if we pretend that human genome is like the English language, to translate many languages to English. Very Anglocentric of me, I know, but stay with me. If we look at which organisms have high quality translations of each word to many other languages, then we jump down another order of magnitude. So that's the reality of the data that we have. And what I'm arguing is that there's very interesting phenotypes that could be explored in molecular cell types that we may be interested in that we just simply don't have access to as a result of not having this genome list. And yeah, even if we were doing a genome a week, as is the fastest computational tools these days, it's still many weeks before we get to all 10,000 species. So back to this analogy. So are these books the same? The spoiler is yes, they're both the same book. They're Tao Te Ching or The Way, one in Chinese and one in Russian, the language I speak at home. Now let's think about not just these books are the same, but are the cells the same? So instead of a book in one language in two different languages, we have a cell in two different species. And could they be the same cell or the same cell type? So our goal here is to align transcriptomes to non-model organisms and find matching cell types. So let's say we have a reference cell atlas and several cell types from there. We would like to take species without reference genomes and identify cells that are similar to known cell types that were found in the existing species. Now, the issue with these, the analyses is that the current state of the art is they discard data at each individual step. So for example, for let's say 20 million reads in macaque, we start with all of the original data and align to the reference genome. Then this is from RNA-seq data set that I ran ran from macaque. There I saw that about 40% of the reads did not align to the reference genome. Now, if we then only count genes that, or data that wasn't annotated genes, or where we actually saw a word, a gene that had a definition, then we had add an additional 30%, may add an additional 30% of data that is in an unannotated region. Then if we subset to only orthologous genes, so one-to-one orthologs, that may result in an additional 10% of the original data that is in non-orthologous genes. So ultimately, we can get maybe 80% of our original data that's just ignored from in typical analyses. And this is what is typically done for cross-species analyses. What I argue is that we need reference-independent methods that bypass these data discarding steps. And the way we do that is by subsampling words from the sequence data. What's not required is you don't need a genome assembly, don't need that gene annotation, don't need these equivalent gene assignments, so it's friendly to non-model organisms. Now, in Western languages, it can be easy to tell the difference between words because there's white space in between them. In German, it may be more tough if there's a bunch of words smashed together to make a new word. So maybe that analogy doesn't fully hold up. At least in the languages I know of, typically there's a white space to separate one word from the next. Unfortunately, in biology, we don't have that luxury of already knowing what the words are. So what we do instead is use short K-long sequences, or K-mers. So just want to quickly define what a K-mer is for those that may be interested, that may need it. So if we have a sequence ATGG and we're using a K size of 3, so K indicates the length of the sequence we're looking at, then the first K-mer is ATG and the second K-mer is TGG, and so on with a sliding window across the sequence. Now in 2019, there was a paper applying K-mers to single cells by comparing the clustering obtained from gene counts with the typical pipeline for single cell RNA-seq with Cell Ranger. That's what I'm showing on the left here. And on the right, I'm showing similar clustering, similar 2D embedding with a slightly different algorithm from using only K-mers from protein coding transcripts. And what they showed is that you could identify or distinguish cell types from DNA K-mers alone. What we are doing differently is we are using protein K-mers that represent biochemical synonyms, and we do a data downscaling or compression. So what we're introducing is a concept we're terming K-Mer Homology, where it's a transcriptome first reference independent method for cross-species analyses. So what do I mean by biochemical synonyms? First, we use reduced amino acid alphabets. So we do 6-frame translation of RNA, then take only the high quality reading frames with an algorithm I'll show you in future slides. Let's say we get a protein sequence called living, L-I-V-I-N-G. You may remember that L-leucine, I-isoleucine, and V-valine all have very similar molecular weights and biochemical properties. They're all small-ish hydrophobic amino acids. So we can categorize them to all be the same category. If we can put L-I-V-I all in the same category in this implementation, we'll re-encode to the letter E. And then the final letters N and G have different biochemical properties. What that means is that the protein synonyms are implicitly encoded. So EECB represents living, the original sequence. It also represents any of LIVI in those 4 positions. So we now have protein synonyms, much like what we could look up in a thesaurus for word synonyms. We next scale down the data size using min-hashed based uniform subsampling. So I'm happy to go into the details of how min-hash works. It's a very interesting, I think it's a really interesting algorithm, computer science trick. We take millions of biochemical synonyms, And we compress the sequence data with MinHash by uniformly subsampling the data. We then get a bag of words, which we're actually representing as a bag of numbers because computers like numbers better. And the combination of these two biochemical synonyms and scaling down the data size is using biological knowledge in the first aspect, plus computer science tricks to then create K-Mer homology. And biochemical synonyms were introduced, almost, what would that be, 60 years ago, for when people were first studying protein sequences, and were recently used for identifying orthologous genes. And the MinHash-based subsampling was originally developed for metagenomics, and I've repurposed those tools for single-cell RNA-seq. So far I've told you about a bunch of hammers. I like building hammers, and then I go searching for nails to apply them to. And there's something to be said by building a really good hammer, I think, and then applying it to a new nail. So I probably don't need to tell you this, but one of the hammers I've become interested in, I probably don't need to tell you about the importance of zoonotic diseases, but as you may know, bats are thought to be the viral reservoir for SARS-CoV-2, the virus behind the COVID-19 pandemic. And zoonotic diseases are a reservoir of potential threats to human health. So studying cross-species analyses, studying single cells across species are an opportunity to understand human health in COVID-19. So, for example, we are lucky to have access to single-cell RNA-seq data sets from the bat behind SARS-CoV-2, from the mouse, a single-cell atlas of a couple dozen organs, and lung cell atlas from human. Now, our sinicus genome doesn't contain key immune genes and other genes that are important for cell type determination. So we do need transcriptome first methods to compare across species. So here's some questions we have. Can we really predict the protein coding reading frame? And can we align transcriptomes from species diverged 100 million years? So we built a tool called Orpheum to translate the sequences from RNA to protein. Now that is part of the central dogma, and it sounds super simple. And of course, you can always take all possible reading frames, but the reality is that about half of those reading frames are not likely. They have not been observed previously in a related species. So what we do is we take the input reads, we perform 6-frame translation, discarding all the reading frames of stop codons. Now we have a bunch of reading frames, and we decompose each reading frame into an amino acid kaa-mers. By kaa-mers, I mean I'm representing specifically the amino acid sequence length. And then we check how many of the kaa-mers are represented in the proteome database. So this proteome database does not need to be from the reference species, the same species. It can be from a species that is diverged. So that's part of the beauty of using proteins, is that the genetic differences is somewhat masked by using the protein sequence. So then we can say, okay, only these 2 reading frames are actually accepted, or maybe 1 reading frame is accepted from this particular read. And this is a way of discarding the clearly unlikely reading frames. So we tested this on evolutionary distances using simulated human RNA-seq reads and a variety of divergent proteins to say thumbs up, this is a protein coding frame or not. Now, Orpheum Translate does have high classification power. So what I'm showing here is the receiver operating characteristic ROC AUC value on the y-axis versus the K-mer size using a protein alphabet on the x-axis. So each color or line represents using a proteome from the exact same species, human, a closely related species, primates, so within about 50 million years, and then rodents are at about 90 million years diverged. And what we see is that while the classification power does is not as perfect as it is with human, using other species, still using rodents, you're still able to get most of the correct protein coding reading frames. And what's interesting is there's a sweet spot of where this is most accurate with a K-mer size of 8 for the translation step. So back to our goal. So far, I've just told you about cross-species translation. Now let's talk about the actual cell type alignment. So we'll take a reference cell atlas from mouse, built on the cell types in mouse, and match to mouse, human, and bat. When I say the mouse, we built a cell type database in one mouse and then checked the cell type assignments in a different independent mouse that is genetically distinct. So yeah, different mice, it's not on the same one. So across mice, from the query mouse in blue to the database mouse in white and pink, I'm showing a Sankey diagram, which is originally developed for engineering aspects of heat transfer. And what we're looking for is where the cells, let's say the capillary cells, have a strong single line from capillary cells in the blue query mouse to the capillary cells in the database mouse. And what we're looking for is not too many crossovers. We see that this is working quite well within species. And if we add other species, such as from human to mouse and bat to mouse, and we are also working on quantifying this, so don't worry if you're looking for scoring, we're working on that as well. But when we query with human or query with bat, is that the monocytes do seem to be dominating the signal. So we are still investigating exactly how to manage this, but what we do notice is that the monocytes simply have many K-mers, many distinct sequences expressed in them compared to the other cell types. They are a more pluripotent cell type, which do tend to express a larger diversity of genes, but we're still figuring out how to manage that. And I think of what else I want to say about that. Yeah, we're still figuring it out. And I'm happy to hear any feedback that you have about this. But yeah, it's like the monocytes with so many K-mers have a bit of a gravitational pull that they're pulling cells that aren't quite like them to them, even though just because of the sheer amount of K-mers they express. I will briefly mention this. So if we look at the K-mers, despite some of this monocyte gravitational pull, we can still look at the K-mers that were used to assign a particular cell to a cell type, which were returning diagnostic K-mers. So K-mers that were that helped to assign a cell to, say, a capillary or alveolar epithelial type 2 cell. So these are K-mers that were expressed in the query cell and found in the database. So there's a match here. That diagnostic K-mer could be not in the reference assembly or in the reference assembly, not in an annotated gene, in an annotated gene, not in an orthologous gene, or in a one-to-one ortholog. What I'm pointing out is these first three categories, not in a reference assembly, unannotated genomic region, and non-orthologous genes. These are the categories of K-mers that would typically be discarded or ignored by traditional methods. And what I wanted to point out is that for the bat, the majority of diagnostic K-mers for each of the cell types would have been ignored. So this is showing that we're getting more sequences, more genes than you'd get with a typical analysis. So as a summary, K-Mer Homology enables building reference cell atlases and hard to study organisms. And we do this by using approximate per word translations aided by reduced amino acid alphabets. And this allows us to match cell types across species. Some of the key tools we built for K-Mer Homology with great help from Pranati Ramori at Cz Biohub. The first tool I talked about, Orpheum, for translating RNA to protein and for the likely protein coding sequence. I didn't talk about it, but the big tool underlying this is called Sour Mash, which we have been working with people at UC Davis on and contributing to it. Finally, BAM to FASTA for getting individual cells FASTA FASTQ files from the 10x Genomics BAM output data. And it is an alignment file, but you can really use any genome to align it. It just, the barcode demultiplexing is its own issue. So these tools we have assembled into the NF Core kmermaid pipeline, and it is under construction. I'm happy to address any issues. I would love for you to try it out on your own data and let me know what you run into, what kind of edge cases you find. And finally, predict orthologs, which was a pipeline I wrote to look up given protein or RNA sequence, look up what the potential sequences could, definitions could be, annotations could be. So brief acknowledgements of the people who worked on the data from the K-Mer crew at Cz Biohub. Again, I mentioned at UC Davis, I've been working with Titus Brown, Tessa Pierce, and Louise Erber, who have been very generous in helping with this project. I want to also acknowledge the people who are generous to share the data. And that's all I have for today. So thank you very much. And I would love to take any questions you may have.
Thank you very much, Olga, for the nice presentation. I will actually allow people to unmute themselves and ask some questions. Oh, maybe. Okay, Sven has a question.
Yeah, thanks, Olga, for the talk. I'd have a question for your orpheum approach. I hope I kind of spelled the name of the tool right. So obviously you used transcriptome data, yet at some point you need to make use of protein databases. So I wonder how the scenario would look like, or how it would compare to the transcriptome approach if you had initially proteome data and you would not need to do the 6-frame translation of the transcriptome data, but could directly compare, so to say, the unknown proteome to the related proteome in the database. Have you considered that, or is that something that is
not? That would be, I have thought about that a bit, and that would be great. Yeah, if you, let's say, had mass spec data. I always forget it's spectrometry or spectroscopy. Sorry, mass spec people. If you had mass spec data, spectrometry, thank you. If you have mass spec data from a few different organisms and would like to compare them, yeah, I think the reduced amino acid approach can definitely help to bypass the need for doing an ortholog matching. And kmermaid does have that possibility of just providing protein FASTAs to then create the K-Mer signatures, as they're called. So the subsampling of K-Mers per each sample. So yeah, so that'd be great to try out. I think that's super interesting.
It would be very interesting to see which approach has better sensitivity, specificity, and okay.
Yeah.
There's another question by Kevin.
Hi, yeah, thanks for the talk. Okay, really interesting stuff. I was actually wondering about the issue that you mentioned with the monocytes, that they make up such a great amount of the assigned cell types. So your assumption is that it's caused by that they have too many K-Mers. Couldn't you just subsample those K-Mers or normalize it so that every cell type has the same amount of K-Mers?
Sure, we can do that. The way that it runs into problems of the way we do the subsampling with MinHash. The way we do the downscaling with MinHash is that when we downscale, it guarantees that if you have a query cell, all the K-Mers that are subsampled from the query cell will be in the database. And with MinHashing, there's two different options for subsampling K-Mers. You can do either a fractional downsampling, and we do a 10%. Take 10% of the K-Mers, or you can do a flat rate. So it's kind of like with a mail, you can either pay by weight or you can have a flat rate.
Okay.
And in my observations, when we do a flat rate, the classification metrics aren't as good as if we do a scaling. Partly is that for the K-Mers that are basically on the border between if they would be included, but excuse me, at the scaling rate or the flat rate. There's no theoretical guarantee those K-mers will be present from the query to the database. The way we get around that is we remove things like K-mers that are in house. Sorry, I don't like the term housekeeping. Constitutive genes, and so ribosomal, mitochondrial, those kinds of genes. We also remove K-mers that are present in 80% of the cell types. So that's another constitutive gene approach. I think there may be some specific sequences in monocytes or relative to pluripotency that we may want to remove that are more related to, say, cell proliferation than they are. That's more of a cellular state versus type, more of a cellular state of proliferation rather than the cell type. So I think I'm happy removing K-mers that are and pretending they were never seen across all data sets, but I worry about doing a downsampling of K-mers that doesn't respect the theoretical guarantees of min hashing.
Okay. Yeah, I was more thinking about you could downsample several times and then repeat your analysis, and then, I mean, if you miss some cell types, you will miss them some other times. And those cell types that are actually truly monocytes or orthologous cell types, they should be catched, but I guess it's not that easy.
Yeah, I mean, part of what's nice about min hashing that I really enjoy is it's more of a computer science trick, is it's very fast. And yes, it could certainly be more accurate, and maybe that's the expense we pay. But the act of min hashing is that for each cell or cell type, you have a bag of words. And if your bag of words from your query cell matches your database cell type, well, you have a match. And doing many of those resamplings, of course, this would be, much more powerful as like a deep neural network, machine learning kind of thing. But I am a simple person. I like using simple methods. And this is a simple method that, despite its flaws, does provide insight.
Cool, thank you.
What's a good K? I mean, can you run with a bad choice of K? Can you run into computational issues that you have?
Yeah, so I do have a slide about this. So the K-mer size of 21 is a commonly used K size for a DNA that's unique in many genomes. So if you do the log math of that, so if your DNA alphabet size, so a sigma here is the alphabet size, so sigma to the K is the total number of possible K-mers at that particular size. So K, your alphabet size for DNA is 4, but for amino acid sequences, protein we have 20, and the Dayhoff alphabet, which I'm happy to show, has a 6-letter alphabet. So to find the ideal or equivalent K size, you can do the log math here to take the ceiling of, oh, I forgot to show the, you could take the log of this to find your ideal K size for a particular alphabet. And for protein, that ends up being about 10 amino acid long. And for the Dayhoff alphabet, it's at 17 amino acids long. So that's what I would just say is the minimum K size for those particular alphabets to represent the same diversity of sequences as is seen for a DNA K size of 21.
So, and what would be as you say, it's a minimal suggestion, so to say, but what would be the disadvantages if you go larger with the K?
If you go larger, it will be more specific, so it may end up being more species specific rather than generic. So what's interesting about the tools from the metagenomics field is that they're many cases trying to use longer K sizes to distinguish between species or strain variation, for example. So they're trying to make things that are very similar farther apart, like make their distance farther apart so you can distinguish them. Here, I'm doing the opposite. I'm taking things that are farther apart, so things that are literally in different species, and using protein K-mers to pull them together. So with a longer K size, they may not come together quite as well.
Okay, interesting. Thanks.
Yeah, these are great questions. Thank you. Sorry, Giselle, I interrupted you.
Yeah, no worries. Yeah, I also have a question regarding, I mean, you showed before that you used also the K-mers to predict which species that was. And I was wondering for like these methods, did you use machine learning as well for that, or like what? And if you did, like what? How do you construct the embeddings for training a model with the K-mers?
Yeah, I didn't use machine learning in the classical sense of training a particular like a linear regression model. That certainly could be done with the K-mers themselves, and maybe would be useful. But yeah, we really just built cell type databases using a data structure called a sequence bloom tree for each cell type. So we merged all the K-mer signatures from cells of a single cell type and built that into one signature, merge those into one signature, and then use that to then look up the cell types.
Okay, yeah, thanks. Yeah, I think John Ho also has a question.
Hi, hi Olga. Thanks for the really cool talk. I like how you use a bit of the NLP tools to incorporate the synonymous translations for the protein to look for the K-mers. So that's really cool. My question is, I understand for this pipeline at least, right? So you use you limit it to one-to-one orthologs. But do you think, or are you aware of how much you're missing? Because when you're comparing between species, you're trying to diagnose between them, right? For example, a lot of missing genes or novel genes, for example, or even paralogs and so on. So do you think you're missing a lot? Or is there any way to overcome this?
Sure. Yeah, so when I describe the one-to-one orthologs, that is more to describe the current state of the art. We are considering all the data. We are considering sequences that could be part of paralog switching events and one-to-many, many-to-many homologs. So using the K-mers, this basically encodes a fragment of an orthologous gene. So we haven't looked into which paralog events we could be missing or could be looking into, but that's a really good point. I think some of the what additional information we're capturing with the K-mers regarding multi-mapped genes or one-to-many, many-to-many orthologs could be really interesting. And that's a bit of what's I haven't because if you're not in the orthology field, then talking about one-to-many and many to orthologs and paralogs can get confusing. So this talk was very simplified, kind of glossed over that topic completely, but there's like consortiums about this. So it's it's not a small topic, but some of these genes that I'm describing here as non-orthologous are simply just not one-to-one orthologs. That was mostly to streamline the message, but I agree with you that it's not quite as representative of the reality of genetic drift and gene duplication and so on.
Yep, thanks a lot.
Sure, hope that answered your question.
Okay, so far I cannot see any other questions.
I might have a question for your organization. It's probably not a scientific question, so
Sure.
Since you mentioned these platforms that you have at the Chan Zuckerberg, and you're in the data science platform, and there's genomics as well. You mentioned. Can you? You mentioned.
Yeah.
It, I just don't recall it anymore.
Yeah. Let me just show this slide again. Yeah, the platforms include data sciences, genomics, so that's genomics and sequencing, engineering, so that's like Fab Lab, physical engineering, and imaging.
Okay, and how's the data science platform connected to the genomics platform? Is there any interplay with respect to data management? To I guess, I mean, doing NF core pipelines. Of course, I'd like to ask, do you have any plans on automation for the data that is directly generated in the genomics platform? How is that working?
Yeah, we would love to get there. We're not there quite yet. We still fire off our pipelines manually, and we're working on getting more NF core pipelines in use at Cz Biohub, so we don't have like a bioinformatics core, as may imagine. It's more data sciences people will help out with particular projects, or make a few tools to get some aspects done. But we're currently going through a transition phase to work on having a centralized database called a data hub to really aggregate all our data together, for laboratory information management system, LIMS systems, metadata tracking, sample tracking, and all that. So all of that is in the works, but is not currently implemented.
Okay. And do the platforms also, I mean, that's primarily as a data science platform, I mean, you have your own research projects, but do the platform also serve like the other platforms in a way with services, or is that every platform works on independent research projects, or how is it?
So I'd say the genomics platform does provide a good amount of services to people within Biohub and also the CZB investigators. The data sciences team, we have some independent projects, and then that are either with people at Biohub, or I also work with some of the investigators. But yeah, it's a mix, and it really depends on the platform itself, on how much of a service provider it is.
Okay. I'm obviously asking these questions because I love to learn from other sides, because we're continuously dealing with reorganization of platforms, core facilities, and all that stuff. So it's interesting to see. And do you need to, if you want to do research projects, then probably do you need to apply for funding, or is that just there in the Biohub, or?
Oh, I have not had to apply for funding,
okay. So
but I know others who have applied for grants from the Bill and Melinda Gates Foundation, and other private donors, because some of the, so Biohub does not accept federal funding, but only private donations.
Sven, can I just briefly interrupt? Since we are moving away a bit from the talk, I think I will interrupt the live stream. And if you have more questions for Olga's talk, I think we can also contact her probably on Slack or anytime, or write her, and then we can continue.